our topic today is, can we quantify soil biological activity without a lab? And we have Dr. Amy Schmidt and Karla, her graduate research assistant. So welcome, Amy, and you can introduce yourself and introduce Karla and start your presentation when you're ready, and I'll stop sharing. So welcome.
All right, thanks, Paul. So I'm Amy Schmidt. I'm at the University of Nebraska-Lincoln. Karla Melgar is a graduate student who's been working with me for a little over a year and a half now. She's towards the end of her program in the Biological Systems Engineering Department, working on a master's degree. And so she's going to talk about some of the work she did for her thesis, and centered that around looking at ways to quantify or visually represent soil biological activity in a field setting. And so I'll let her take over, and I will help out as needed. But Karla, please feel free to go ahead.
Yeah, thank you. So like Amy said, my project as a graduate student started out as trying different organic amendments for corn production, and then it just started arising a lot of questions about what matters besides chemical properties in growing corn. So we started searching for other ways that we could measure soil health, and few of them were physical properties, but also how all of that is affected by biological activity in the soil. So just to give you a little background on it, the idea was to identify the things that make a fertile soil. And so we wanted to study nutrient composition and soil structure and microbial activity, and how all of these connects to each other when we are using especially organic amendments, because we know they're not available right when you put them in the soil. So I wanted to study a bit more of what needs to happen in the soil before those nutrients are available for the plants. And so the idea was to see microbial activity, but and with that, all the roles that microbes have in the soil and organisms like arthropods and all of that, and how they shape the soil structure to make it ideal for plant growth, and how to make it resilient and prevent water contamination, especially with applying manure. So the problem with that was, first of all, I'm not a microbiologist, and I don't think I could learn a lot about that for my master. So it was a matter of how can we measure microbial activity without having all the equipment that we need for the lab and all this knowledge that probably someone with a degree in biology has. I didn't have that background, but I did know that we could go out in the field and test biological properties somehow. And while I was doing that, I was kind of going through what methods there are out there to test microbial activity. And these are just a few examples of it. There are some field sampling kits. I've never used them, but they're around, I think that one over there is around a couple $100. And then there's also soil incubation that you can do, and that kind of indirectly measures microbial activity. We tried that out in our lab, and the equipment is not something out of this world, but it does take a little bit of being very precise and having an incubator, for example, and that's not something that you can have like at a field station or something. So it was valuable to see it, and it's a good thing to measure if you have the things for doing it, but it takes a little bit of practice and reading. And then there's also images that you can take with a microscope that allow you to see all these things that are in the soil, but then you also need to know what you're looking at. So just by looking at this image, I know there's something in that soil, but I don't really know what's in it. And that's something that I feel like for most of the people who don't have a background in microbiology would be very hard to understand. So we wanted to create a method for evaluating soil health through the Soil Undies demonstration. That's very popular, especially as a visual tool to show farmers the difference between soil management practices. I've seen it done with tillage and no tillage or cover crops. And we wanted to test this out with organic matter, organic aggregation in the soil. And the reason why this is a good tool to use is because cotton fabric is made out of the same components as some of the organic residues like leaves and sticks and corn stalks and everything. It's degraded on the kind of the same way and with the same microorganisms that cotton fabric is. So then we just came up with the question, so how do we measure this in numbers? This demonstration is very popular, like I said, but usually it's just, you bury a piece of cotton fabric or a piece of underwear and then you show it to people. And that has an impact, but we wanted to put numbers on it and see if we could actually evaluate different management practices with it. And so with that, we needed to create something that was standardized and proven to be an actual tool that can be used and that the results are applicable by others. And, that we had the right tool so that other people and extension offices could use that in other types of research, and that we would get accurate enough results to know that there are changes happening in the soil. So to set up this experiment, the first set of cotton fabric that we started trying out was 12 by 12 inch, 100% cotton fabric. And they were placed inside these mesh bags that you see here, just so that we don't lose them when we put them in the field. But they still allow for the fabric to have direct contact with the soil. And then those were buried at a 2 to 5 inch depth of soil. And then in the field, we put different rates of manure and compare that to control treatments that had no fertilizers. And for the first experiment, I was taking those samples out every week and just taking pictures. And so the picture process, I think just getting there to a part where you could have a good picture of it was a bit challenging. You had to set up some lights and mark down an area in the back so you have a pre-measured area. And then, so here are some demonstrations of the different treatments that we tried on the same week. So this were all taken on the same week. And this picture in the middle is actually a little bit edited because when you get it out of the field, it looks like the manure only picture. So it's a lot of stains and a lot of dirt. And then, so you kind of need to do a little bit of a process that I will explain a little bit more later. But we wanted to test out different photo editing software. So one of them was ImageJ, which is very commonly used for microbial experiments or microscopic images, because what it does is it gives you an amount of pixels that are in a picture depending on the color. So in this picture right here, it will tell you how many pixels are white, how many pixels are gray, and it kind of ignores what's in between or makes it, if it's a dark gray, then it will count it as black or something like that. And there's something similar with Photoshop. Here, this image, you can select around the picture and the program will tell you how many pixels correspond to a white, for example, white area of the picture, or how many pixels are black. So they both work in a similar way. The difference is ImageJ is free, and so you can download it. It's a little bit, for me, I think it's a little bit more complicated to use, while Photoshop, you can find a lot of tutorials on it and it's pretty straightforward. And so we compare those pictures with some of the fabric that we took out and we put it on the grid. And we hand counted how many squares were inside that grid. And so that way we compared hand counts to degradation that we obtained from ImageJ in Photoshop. So if you can see here in the graphic, it's comparing Photoshop versus ImageJ versus the hand count on the x-axis. So they all were pretty consistent in determining the degradation proportion. And then this little dot that says black light was us trying out different sources of light so that we can get a better contrast on the picture from the white and the holes in the fabric. And then this picture on the right was a different experiment, but with this one, we counted hand count, Photoshop, ImageJ, and the fabric was taken out of the field every 3 to 4 weeks. And still you can see the results are pretty consistent. So the different colors are different methods and they're all pretty much the same. So I can say that the 3 of the methods are pretty good at giving you a degradation rate. Hand count, they're a good way to measure that the degradation that you're getting is right, but it takes a lot more time to get a hand count estimation, and you have to be more focused than you think you do because you miscount 1 square and it just messes up everything. And this is just a graphic of an application that we gave to this. So when we were testing out manure versus control and using the results that I got from Photoshop, we actually were able to get a degradation percentage that was pretty accurate. Here you can see that manure had a higher degradation in time than control plots, the pink ones. And so, like I was telling you, some of the challenges that we encountered with this was the fact that it is white cotton fabric and it is going to get stained with soil. And when you put it on the program will not tell you this is fabric, it's just dirty. The program is going to tell you this is white or this is black. So what you want to do is kind of like edit through the picture and make sure that what you're seeing is either a hole or is still a piece of fabric. And so that way the program will be able to tell you more accurately that that is a piece of fabric. So that took a little bit of manual adjusting of the colors. It didn't take more than 5 minutes probably for each picture. So it's not that hard, but you do need to be a little bit careful so that you're not mistaking a stain with a hole in the fabric. And the same thing with ImageJ, but the way of adjusting things here is a bit different. In Photoshop, you have like a little pointer that you can move around and color the picture. With ImageJ, you need to select areas of color and it will make it a different color by itself, but it's still something that you need to select manually. And so we were trying to find ways to standardize the process and make it ideal so that we don't have to intervene so much with editing the different parts of the picture. One of the undergrad students that's working with us found this little box that's specifically for taking pictures of objects. They have it on Amazon, and I think that would be a great tool to incorporate with this. It has like LED lights and a white background that I think you can switch to a black background. And well, the idea of this is just use whatever you have available so you can easily adapt that with a cardboard box and some LED lights, and just trying to make sure that you are always taking the picture on the same place and in the same angle every time so you can have comparable measurements. And he actually left a video here that he was trying to implement the same basic commands with Python. I'm not an expert in Python, I've never used it, but he tried putting a code in Python, and what it does, it's assigns number to each category of color in the image. So when you take an image, it will tell you how many of pixels of a certain color there are. And I think the advantage with this would be that you can name, for example, a dark black color is number 0. So the program will tell you how many zeros are in the picture, and you can make that more automatic once you figure out how to code it perfectly. But that is something that can be created as a cell phone app, and it's pretty easy to use. You don't need to edit so much. And so he did a few examples of it, and turns out that the results that you can get with Python are pretty similar to ImageJ and to Photoshop and manual counts. So that's a video of the process of editing. So looking forward, I think that would be a very good thing to do if we wanted to make this process a bit easier. And you can do it with your phone and kind of like avoid editing so much of the picture in a laptop. So I guess in conclusion, I can say that the results are replicable with different methods. So even we explored Photoshop and ImageJ, but I think that could be easily done with a lot of photo editing softwares. And we actually had, I don't know if Gary is here, but there was a group of people from this extension office that in Julian, Nebraska, and they were trying to do some of the same experiment using cover crops. And I was helping them out, just like going through the steps to get the degradation estimation. And it was pretty easy for them, I would say. And so it's something that people can replicate, and it's a good way to show people. I think it has a lot of impact when you see a piece of fabric under different management methods, and you see how much one of them is degraded versus the other one. And yeah, very minimal equipment is required. We only needed a camera. We actually used a tablet camera. It was nothing professional or out of this world. And then just a background, a dark background that you can use as a contrast. And yeah, just the photo editing software. And so, yeah, I think it gives pretty good results. It's very consistent with either of the methods that we tried. And yeah, so I don't know if you have anything to add, Amy.
I would just say that, yeah, the project kind of started out as, can we figure out a way to use that fabric degradation process that we've all seen with the Soil Your Undies experiment to come up with a quantified degradation of that fabric. And I think the students did a really good job working through several different methods. Of course, we had a grid for hand counting and then comparing that to different visual photographs and images. And I think we still have a ways to go on it, but it's encouraging.